all these things are like building blocks. The whole story's been built. I'm just kind of giving you a trailer. Hey, this is all. But you're not talking about the immune response. So there, everything will come together. Now, let's talk of MHC before we jump onto the T cell. And I'm going to talk about genes. What is an MHC? Again, what is a variability? When is it that the T cell would not respond to an antigen? What are the different types of MHC and so a little bit of genes, a little bit of genetics again, but only related to T cells. Okay? Now, you all know, especially in terms of chromosome 6, there's a long arm and short arm. In chromosome 6, on the short arm, there is a region we call human lymphocyte antigen. We call human lymphocyte antigen because it's present on the lymphocyte, which is called HLA, right? So basically, MHC is a misnomer. Major histocompatibility complex was discovered in animals first. But the corresponding thing in humans is HLA. So if you're going to go for your tissue typing, you want to donate a kidney, they're going to do HLA typing, which is an MHC. I mean, just like, because it's been so widely used that we still use it. So in this short arm of HLA region, there are 3 classes, class 1, class 2, and class 3. So this is a gene map of human leukocyte antigen, right? And these are those specific series over here that when you do blood matching, we don't do, blood matching is not at that level. It's like a carbohydrate level. We'll do that on Monday. So this is more of a superficial thing. But at the lower level, what normally happens is that class 1 will have all these genes, B, C, E, A, G. So if you were to run for those of you who have seen the report for the HLA typing, so they're going to give you a number. So you will have a number of all these things. They will give you a number of your DP, DM, D. So they will give you a very assigned number. This is your tissue number. Like I told you, the barcode for the computer. Exactly. Then they're going to match it up with the other and see if there are similarities or disparities. This is a tissue map. And remember, when we say some of them can evoke a very strong reaction, and some of them may not evoke a small, strong reaction. So we have to make do. So you have to kind of get around that. Hey, let's do that. Okay? So class 2 has DP, DM, DQ, DR. Don't remember that. Class 3, because we normally don't talk about that. We do talk about class 1, B, C, E, A, G, F. So these are the major HLA antigen region that we do for gene mapping especially for this part I'm going to take you through. Now, let's see what happens. Okay, this is a host cell, okay? And this host cell then basically takes in a protein antigen, right? So the protein antigen has to go over here. This has to be broken apart. That's why antigens for antigenicity have to be hydrolyzed, broken apart, degradable. So it breaks up into 3 peptides. It breaks up the 3 peptides, right? So the protein is broken up, and then this basically needs to be presented to T cell on MHC molecule. But remember, this MHC is basically HLA. We use MHC because we get so used to this term that we still kind of take it as HLA. So let's take it as an HLA as well. So your cell produces MHC molecule, takes a chunk of this epitope, we call it epitope, and sticks it out to the cell membrane. Right? Now, any cell that takes an antigen, breaks it up, and presents, so this cell basically is an antigen presenting cell. This has a capability of presenting an antigen. Can red blood cell present an antigen? No, because they don't have a nucleus. So they cannot make MHC. But this one particular, any host cell will present, and guess what? There's only one protein, what do you call a meat eater or person who's going to come and look at it through what receptors? What receptor is there is going to look at MSC? T cell receptor. So don't get confused, MSC versus MSC, that doesn't happen. So MSC, plate, protein, T cell comes, looks at it, TCR, but this is only a first handshake. Just want to handshake that T cell. But remember, T cell as a rule would not act unless it needs at least more two forms of identification before it kind of gets going. So it doesn't really you just want to hold on to two hands. Or I usually say T cell will see an antigen with two eyes. So that is only one eye. It's presented, fair enough, interaction happens, but T cell would not do anything because it needs more information because it is a very major cell. It's a decision maker in the body. It's like whatever, we had that, the incredible guy. So he's a decision maker there. So it has to be very, very careful. But this is the first step, the use of MSC molecule. But remember that MSC molecule, T cell cannot come inside and look around. That's a problem with T cell. T cell is not going to dig out and search your pockets. It's not going to do that. Whatever is expressed. So these cells see external. Immune cells, blind. As you say, they're blind. They will only see when expressed. If you keep this antigen inside, nothing. There's nothing you can do. Okay? So this is called antigen presentation. T cell sees it. And then this step we call antigen presentation and antigen recognition of the cell. So these are the terms that we use. And now TCR hooks up with MHC to see that part. But I also told you it has to be a smaller chunk. It cannot take a big part. Okay? Yes? So this ability of T cell to only eat on a plate or only see the antigen when it's present MSC, we call it MSC restriction. That's what we say, restriction. So it's already restricted. That's the term we use. We call MSC restriction of T cell responses. There is no MSC restriction for B cell responses. So these are type of questions that you will get. I mean, these are the concept questions that you may wonder what is it that I'm going to ask you. So these are concept. There's no MSC restriction for B cell. There is an MSC restriction for T cell. Reason is because T cell exclusively responds to protein and small peptides. And interestingly, many microbes are proteins. So in order for you to fight microbes, which we'll discuss later, you need T cells. If you have no T cells, for example, what happens in HIV? Virus kills your T cell. So what will happen? You get infection and die. Because you don't have T cells. That's how the AIDS people die. Because they have no T cell, no fights put up. The critical role in response to every, and then again, the interact with antigen inside the cell. So remember, that's another thing, that the bacteria, the whole chunk of protein has to be processed inside the cell and broken apart, very small chunk of amino acid that fits on MSC, that fits on TCR, because there's a capability of both of them. Okay, so that's what we call MSC restriction. Okay, I already told you MSC basically is human leukocyte. It is inherited as a unit that you saw the unit sequence, and every vertebrate has MSC having multiple genes. You saw all those genes that we have. Okay, now let's see what's happening at the junction. Now you can see from here, this is the host cell that you saw, and this is a T cell. So host cell then presents MSC. This is like an MSC, and there is a groove on MSC. That it is a groove there. It's a special, like a key thing. No, and it has a TCR as a corresponding groove, and they both want to see that peptide. So it has to be that much of a peptide that snugly fits over here. If you're going to offer him a bigger chunk, it's not going to fit. It's going to be loose. So that is very tight thing. The cell has to break it apart into small chunk, put it up over there, snugly fits like a Velcro, and then the TCR will come, and that they start talking. They would not talk. There's no deal between T cell and the antigen presenting cell unless and until both of them snugly fit this peptide over here. That's how the negotiation will begin. Okay, now so far I just said T cells, but again, there are many different types of T cells. In B cell, I just told you memory cell, plasma cells. Pretty much it. Okay? But in T cell, I'm going to give you a list of T cells. Based upon function, you can memorize in different way. But what we call those T cells as subsets. So they are subsets of T cell. Right? But what you notice was that the function of MHC is to make sure that the protein is broken inside the cell and presented to appropriate TCR. So what it means is that if you take a protein, for example, and your cell breaks it apart into A, B, C, right? So there is a special T cell which is designed that holds a TCR that fits with A, B, C. Not every T cell will do that. So that's a specificity of a T cell as well, as we had specificity for the BCR based upon variable portion. Likewise, remember when I told you that the TCR also has that glove portion, so it's going to put on that glove. So that's what we call appropriate TCR. Okay, let's take example of two important T cells, CD4 T cell and CD8 T cell, both look alike. Both of them will have a TCR. So you can see over here, TCR, 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 right? But like B cell, they have a co-receptor. We had 4 co-receptors, I told you the BCR, 4 co-pilots, that they just have one in this one, a very strong one. The co-receptor for, well, let me go back. This is called CD8 cell because the co-receptor is CD8. And this is called CD4 T cell because the co-receptor is CD4. So these cells are given a specific subtype based upon the co-receptor. Okay? All right. Now, I've been talking about antigen presenting cells. So these antigen presenting cells, for example, are going to present, this cell has a capability of taking MHC class 1, put on a peptide, and present to CD8. Now I'm going to tell you another important thing, and there's a mnemonic for that, right? That some of the cells have ability to present a peptide in context with MHC class 1. Some of the cells have the capability to present the peptide in context of class 2, right? Okay? So 2 plus 2, 4. So remember that 2 will only present to 4. If you remember this, the other one is an opposite for that. And 1 can only bind with CD8. So if I was to ask you, what type of MHC is CD8 going to bind? So it's MHC class 1. And what type of MHC CD4 is going to bind? It is class 2. Okay? The other names for these cells is that CD4 T cells have many functions. Let me give you one function, helper. So you call them helper cells. Okay, but that's not that simple. And CD8 is a killer. So we call them killer cells. So, I mean, these terms I'm going to change around. Now, we talked about the class 1 and class 2. And remember that when it is presented onto the cell surface, so these protein, you can see class 1 is this one, and class 2. So when I say MHC class 1, so those some will pick up proteins from class 2, pick up protein from class 1. What I want you to know for this one is the DP, DQ, DR, you can see from here, they all look similar. Most of them have alpha beta chain, very important for class 2, that regardless of subtle details, they all have an alpha beta chain, class 2. Class 1, again, has ABC component, but they have all beta 2 microglobulin. So that's something you have to remember. It's in the book, beta 2 microglobulin. So that is an important player for class 1. And if you don't have beta 2 microglobulin, some of the patients don't have, then you cannot possibly present an antigen in context with class 1 because beta 2 microglobulin is absent for class 2. That is the difference basically in terms of the class 1 and class 2 in terms of the gene expressed on the cell surface. So our ability for class 1 is different. They are genetically determined, and then again the differences that we have in terms of MHC are coming from, we call that polygenicity, because your genes will be picked up at different levels, HLA-B, HLA-C. So all these things are there in terms of expression of gene. But remember that every nucleated cell will have MHC class 1. So that's a rule. If I ask you which of the following cell does not have MHC, and I put red blood cell there, red blood cell would not have MHC because it doesn't have a nucleus. Then there are 3 for MHC class 2, we already discussed that, and both of them have 6 different kind of expressions in terms of the choices are DPDQ. So if you go and actually look at the report, you will see that subtle differences in terms of variability that we get from MHC class 1 and 2. Okay, so these are polymorphic. So we have, like, for example, genetic polymorphism in terms of groups, and then there are different versions of alleles we already talked about when they are. So the chances are, since we pick up different class 1, class 2, each one has a different genetic background. So what it means is that bacteria may have the same gene, but we may have different MHC molecules. So our ability to deal with will be all different. Remember I told you the new pharmacy is going to be genetics, because drugs are targeted according to the gene. The reason being, because if antibiotics, for example, are not going to go for bacteria just because I'm going to give penicillin to all of you. That doesn't make sense. Because the reason is because we have polygenicity, genetic polymorphism in terms of genes. So as we have different blood groups, we have different genes. So we have differential ability. That's called personalized medicine. You know, that's something that they are saying within the next 20 years, or I don't know how good that figure is, that you'll see the whole change, and that's where the diversity comes over here. Okay, a couple of more minutes, and you can see from here this is a biochemical structure, but I want you to make sure that you understand these are all superfamily, and this is class 1 molecule, and as you can see from here, all different structures. Cell membrane, CD8 binding site. The most important over here, which is separate, is beta 2 microglobulin. So that's the important part in terms of microglobulin. Okay? And what you see over here, if it's MHC class 1, so it has a very specific groove. So the peptide over here has to snugly fit into those beta sheets of this protein. You can see from here the actual crystallography. But in cartoon-wise, if I was to look at cartoon of MHC class 1, so you have a alpha 1, alpha 2, it has a peptide binding groove, and then it has to properly anchor there and present to T cell. But it can only take 8 to 9 amino acid residues. So you either break it up into small 8 and 9. So MHC is a little bit, 1 is smaller than MHC class 2. So you have to break it apart. That's what we do in vaccination. We take the chunk, put it in 8 to 9 amino acid, and give it to you. So your cell present it to your T cell for a good immune response. So MHC class 1, you see alpha 1, alpha 2, we have these residues, and then it's going to go for a corresponding T cell receptor on the top. Okay, now let me go to the figure part. I think this will be the easier part. So this is going to be the last one today. So you can see difference between MHC class 1 and class 2. In terms of structure, I told you this one has beta 2 microglobulin. That's what I told you difference. As for expression, all nucleated cells. But this one is constitutively, I use this term, presented on cells that have capability of antigen presentation. If I was to ask you, name antigen presenting cells. So these are the cells: dendritic cell, macrophages, B cell, thymic epithelial cells. So these are the cells they express. As far as the size of the protein, they take 8 to 9 amino acids, 12 to 17, so a little bit bigger than that. In terms of what kind of antigen they deal with, they take antigens which are endogenous, that have been catabolized from the cytoplasm within the cell, as compared to exogenous, which are coming, which are again in acid compartments. A little bit of difference, I'll explain that to you when we move along. And then again, MHC class 1 is expressed on CD8 T cells and natural killer cell, whereas MHC class 2 is expressed on T cells. Okay?